It's Jim, the author behind the blog, Reverse Engineering the Origins of SARS-CoV-2. This is going to be part two of the DARPA Defuse proposal. Part one was the background and what led up to it. Part two is going to be who funded it. So, spoiler alert, it was this guy. So, we'll dive right into it. Part one, we ended on the Fauci grilling. So, Congress now has a copy of the DARPA proposal, the DARPA grant in January, February of 2022. And now they're asking Fauci, did you fund it? Because that's essentially what was written in the cover letter by Major Murphy. He is written down here. Let me see if I can find it real quick. As is known, Fauci funded it. So right here in the middle, it's hard to see it. I have this transcripted into my book in chapter 7, I believe. DARPA rejected the proposal because the work was too close to violating the gain-of-function moratorium, despite what Peter Daszak says in the proposal. That work would not. As is known, Dr. Fauci with NIAID did not reject the proposal. The work took place at the WIV and several sites in the U.S. identified in detail in the proposal. So this is written in August of 2021. This is written by Major Joseph Murphy. So this was published in January 2022. So Fauci's finally being asked.
Senator, I really pains me to have to just point out to the American public how absolutely incorrect you are. What came out last night on Project Veritas was a grant that was submitted to DARPA. Then it distorted and said, we funded the grant. We have never seen that grant, and we have never funded that grant. So once again, you are completely and unequivocally incorrect. When you joined the DARPA proposal was a grant that we never saw and we did not fund. So you are incorrect, Senator.
So the word that DARPA grant is doing a lot of work in prepared denial. So you can see his hands shaking too. I want to do it again.
I have to just point out to the American public how absolutely incorrect you are. What came out last night on Project Veritas.
Last night was January 10th, 2020. So this would be January 11th, I guess, 2022. Sorry.
Was a grant that was submitted to DARPA. Then it distorted and said, we funded the grant. We have never.
So we funded the grant was Fauci has obviously read Murphy's letter, and he's seen what Murphy wrote in there. Fauci funded it, and so Fauci's kind of come up with an alibi in 12 hours or less.
Seen that grant?
That grant.
We have never funded that grant.
That grant.
So once again, you are completely and unequivocally incorrect.
So that was his prepared denial, and people took it hook, line, and sinker. So I'm here to prove to you that he did fund it. So first of all, Fauci saying we have never seen that grant or funded that grant is it's we is a strong we. We have seen it. If you talk about NIAID, we have seen it because NIAID was a member on the original DARPA Defuse proposal. This is January 2018. Peter Daszak, basically in the Defuse team of EcoHealth, University of North Carolina, Ralph Baric. Ian Limpke was even on it. Wuhan Institute of Virology. Xizheng Li, she looks familiar in Duke University. Limpha Wang. This is the pre-bid, and they basically are doing musical chairs, trying to find partners to partner up and do this kind of extensive international work. And on the original list was Rocky Mountain Lab. So that's the intramural NIAID biodefense lab in Hamilton, Montana. And Vincent Munster is Tony Fauci's personal, essentially, aerosol specialist. They have a lot of emails to each other during the pandemic. To give you an idea, Vincent Munster gave Tony Fauci a detailed presentation on the origins of COVID on January 27th, 2020. That was 4 days before he was told it was engineered. So Fauci was a member, or NIAID was an original member on the DARPA Defuse grant. So that is essentially a lie when Fauci says we have never seen that grant and we have never funded that grant. They were original member. Now, were they a member in the final submittal? No, they were not. For various unknown reasons, they were removed. They did win 2 of the other projects, which we'll talk about. They were winners, but not with this team. They teamed up with other teams, so it gets really confusing quick, this musical chair game. But that is in essence a bit of a lie by Fauci. So this is how it was funded. So it was sitting underneath their nose the entire time. It was this 900 pages of FOIA material by The Intercept on September 6th of 2021. This is before DARPA Defuse even leaked by Major Murphy. And we'll see why Major Murphy, leaked Defuse here. This is part of my favorite story that, I'm recording this in early 2025, and only now did I get my brain around after writing the book. So here, Major Murphy has written the letter to the Inspector General on August of 2021 with all the DARPA Defuse documents inside of it. And he's declared Fauci as the person that funded it. But he didn't back it up like we'll do here. So what he's done though in this letter, he's followed up on it with an undated letter, and here he doesn't have the date. It's been redacted, or not even filled out, but we can tell when he sent it because you probably saw earlier this week that more official documents linking the NIH and EcoHealth to the Wuhan Institute of Virology were published by The Intercept. So earlier this week is September 6, 2021. That's what he's referencing there. And once he saw Major Murphy, the insider, the whistleblower, the angel, trying to bring some, logic and reason to this pretty messy, contentious debate. Once he saw the 900 pages of NIH grants, he himself said, oh, this is it. This is how it got funded. And none of us knew it, myself included. He's just so far ahead of his time. It took us, us mortals years to catch up because he's an insider. We're outsiders trying to piece this thing together as he threw another puzzle piece out the window and it just slipped right into our nose. So there it was. So this letter was written sometime in September 7th, 8th, 9th, and it was a follow-up to this August 2021 letter. So this is how Fauci funded DARPA Defuse right here. And The Intercept had to sue the NIH to get these documents. They did not want to give them up willingly. But if you go through, which, no one in the right mind wants to go through 900 pages, but you'll see it's essentially Peter Daszak has resubmitted the DARPA Defuse proposal into NIAID grants. It's not, easy to follow, but it's doable if you don't, if I don't lose you here. This is the blog here that outlines how he did it. So here it was. These are the 2 grants. The 600 pages here, the 900 pages here is broken up into right here. So this grant right here was 300 pages. This grant right here was 600 pages. So that's, and once you get your brain around it, it's pretty easy to see how it was done. It was Peter Daszak, in his genius, being a commercial, salesman. He split the grant up, this $14 million grant over several, 3 here in particular that we'll talk about. I just want to focus on these 2. This one, we're going to leave aside. But this 2R01 was essentially what you've heard about being debated by, everyone and their mother when it comes to the lab leak debate. It is the subgrant to the Wuhan Institute of Virology that was awarded in July of 2019. That sounds like an interesting date, doesn't it? But it was bid in October of 2018. So this 2R01 stands for the second renewal, the second round of funding. So it started off as an R01 in 2014, then it's up for renewal in 2019. So then it's renewed and now it becomes 2R01. And the renewal was really weird. Everything kind of stayed the same more or less in the R01, except they added Ralph Baric to it. Ralph Baric of UNC was not on the 1R01, the first round from 2014 to 2019. But he was on the second round of funding and the second round of grant, which was odd because he's a high-dollar scientist on a relatively low-dollar sub-grant to the Wuhan Institute of Virology. It was so odd it came up in testimony. The odd part of the 2019 renewal was the inclusion of Ralph Baric, a high-dollar scientist on a relatively low-dollar sub-grant. It was so odd it came out. Did you have any conversations? This is the committee, the COVID committee to Baric, during his testimony in 2024. Did you have any conversations with Dr. Daszak of EcoHealth regarding the termination? What termination is he talking about here?
Improving greatly, please.
Thank you, Mr. President. U.S. intelligence is saying this week that the coronavirus likely came from a level 4 lab in Wuhan. There's also another report that the NIH under the Obama administration in 2015 gave that lab $3.7 million in a grant. Why would the U.S. give a grant like that to China?
The Obama administration gave them a grant of $3.7 million. I've been hearing about that. And we've instructed that if any grants are going to that area, we're looking at it literally about an hour ago, and also early in the morning. We will end that grant very quickly, but it was granted quite a while ago. They were granted a substantial amount of money.
So this is Trump's first term. I'm recording this in 2025 during the start of his second term, but this was April of 2020. This was the grant that caught everyone's attention. Tony Fauci had money going to the Wuhan Institute of Virology, but it was only part of the story. It wasn't all the story. So that money that was going from Fauci and IID to the Wuhan Institute of Virology was essentially DARPA Defuse money. Just none of us knew it at the time because it took us forever to piece all this together. So that was the grant that's been canceled by Trump and his administration. And Fauci and NID pushed back. They did not want to even cancel it, but they essentially forced him to. And here's Baric's answer to why he's been added to this grant. So Baric, again, was added to the grant in 2018. Let me stress to you, in 2018 is not a random place and time. October 2018, Ralph Baric, Peter Daszak, Linfa Wang, and Xizeng Li, and there's Daniel Anderson in the background, are in Wuhan, having a kickoff meeting for essentially the DARPA Defuse project, which is restarted. So Baric's been added, and Baric was asked, what are you doing on this grant? Baric's answer was not great. I hadn't received any of the money to do anything on that grant yet when the termination notice hit. So he called me, Daszak, and told me the grant had been terminated. And so I knew about it. But in terms of how that would impact my program, a very small component of that grant. When did you get added to the grant? After the first round, so it would have been in the second round. I don't know exactly, can't remember. Going into year 6, that'd be 2019. It would have been going in if year 6 was 2019. That's when I would have been part of it. My role was to study a couple of viruses that the Wuhan Institute of Virology found that they were willing to share with me. So I always viewed that as not number 1 or number 2 on that list, maybe number 5 or number 6 on that list. The shared bat sample was obviously R8TG13 since it fits Baric's profile. Baric was always looking for a genome that was about 25% different than SARS-CoV-2. Or SARS-CoV-1, I'm sorry. This gets really confusing really quick, doesn't it? So real quick here, this is going back into the DARPA Defuse proposal. If I search for 25%, I'm going to find, what Baric wants. This is Baric, in his targeted immune boosting. He wants to find a genome in China or wherever in Southeast Asia that fits this profile he's looking for, 25% diversity from SARS-1. And there, is essentially Baric's mind at work. He wants 25% profile. So it's pretty obvious once you know what to look for. 25% diversity is shown here too. So essentially Baric's creating a novel chimera based on a bunch of a consensus sequence, as he calls it, based upon a bunch of different samples. So the 25% genome he's talking about, he's got this thing called HKU3 SMEX. So that's essentially SARS-CoV-2. That is a SARS-CoV-2 genome in Baric's freezer in 2018. And he's piecing it together. It's just a, it's a placeholder. It's I don't want to get into like the differences of 25% because nucleotide, amino acid, spike only, whole genome. It gets really, really into the weeds. But I'm telling you, point blank, it'll come together later. HKU3 SMX is SARS-CoV-2. The SARS-CoV-2 genome is essentially this one right here. So Baric is slowly assembling this SARS-2 genome over time. This is not nefarious because he basically sees it, as something that he can use to present to the mammalian model as creating an antibody response. He's trying to box in the virus. This 25% different, was terminology he used going way back when. So it fits his profile, I say here. When the COVID committee asked Baric what was in his freezer, he cites up the question. I usually look for sequences. I'll find something interesting, then I'll pursue it. So that's something interesting. Was obviously something like R8TG13 since it fit his profile. So that's Baric has been added to the R01 in 2018, and it was awarded in 2019. So DARPA Defuse is on. The only reason we didn't know it was on, though, was there was no fear and cleavage site in this R01 work, because it's more or less involves Xizeng Li going out there and doing, what's called TA1 work. So TA1 work was, this is the DARPA Defuse BAA, as the bureaucracy calls it, the RFQ, the request for quote, as I call it, in January of 2018. They want DARPA wants the groups like EcoHealth and Baric and Wuhan to break down a TA1 and TA2. TA1 is to go out there and find the animal reservoir, find the hot spot, find the region. So that's just like the R01 work, isn't it? So that's what Peter Daszak has done. He's taken the TA1 out of DARPA Defuse that he lost, and he resubmitted it as the 2R01. So TA1 in DARPA Defuse has been funded in July of 2019. And this is where it gets a little tricky, and this is the interesting part. TA2 is do something about it, develop the scalable approach, and suppress the animal virus in its reservoir, i.e. the bat, and reduce the likelihood of virus transmission to humans, preempt the spillover event. So they now found the virus in the bat, now do something about it, is essentially, so that TA2 has been resubmitted in this NIAID U01. R01 is big money over 5 years. U01 is even bigger money over 5 years. This was 3.2 million. The U01 was 7.5 million. You add those two together, you get what, 10? So, 10.7. So you're getting very close to the 14 million that Peter Daszak originally wanted to submit in the DARPA Defuse proposal. So you remember here, he wanted, what did he want? It's on the cover page, I thought. Yeah, it is. It's somewhere down here. There it is right there. 14.2 million. So we're getting pretty close to the, we're getting pretty close. Once you add this one and it gets there. But they're just focused on these two right now. So after he's got the DARPA Defuse project running, TA1, and i.e. the R01, he's now going forward with the TA2. This is the virology. This is where Baric, genomic sequencing and reverse genetic system really start to shine in the U01. So it was bid in June of 2019, and it was awarded in January 2020. But in the process of bidding it, they have to go through and test it, and they're testing it with Daniel Anderson at the BSL-4. So Daniel Anderson is part of the U01 alongside Ralph Baric, but she was not part of the R01. So Xi Lee with her BSL-2 doing this TA1 work and is pretty, it's pretty benign. She's using Vero cells and humanized mice, which have nothing to do with SARS-CoV-2. But Daniel Anderson is listed alongside Ralph Baric in the DARPA Defuse proposal. So we can see her name down here. This was always the thing that caught my eye. Let me see if we can scroll down. It's just such a big document that'll make your head spin like a top. So here it is right here. This is Ralph Baric right there, right next to Daniel Anderson right here. I can't even highlight it. There's her boss, Lympha Wong. And there's Daniel Anderson right below it. So those three are together. And they've been put back together here in this NIAID U01 grant, along with Lympha Wang. So, and then here's the real tricky part to see. It even referenced Vincent Munster a couple of times in this grant. This is 300 pages. This is 600 pages. Have fun, going through it. But then I think the thing that really, finished this off for me at least, and hopefully for you, is the U01 grant referenced the R0, or referenced the fear and cleavage site. So deep into the draft of the U01 grant, it references the fear and cleavage site. I'll scroll down and show it to you here in a minute. So, and it also referenced Fauci's, what did it reference? Fauci's Rocky Mount Lab and Vincent Munster. So they're back on the team together. So they're off after when they lose DARPA Defuse. They're back on now with Fauci's version of DARPA Defuse. So the Rocky Mount Lab is pretty prevalent in the U01 work. But then this one, just to finish it off, I got it open here. The DARPA Defuse grant covered Southeast Asia essentially. That's kind of, I don't know if he doesn't say Laos in particular, but he says Southeast Asia a couple of times in it. So caves across Southeast Asia and South China is essentially what he's looking for. And that's covered in this R01 grant that was post-pandemic award and date. So that was bid in May of 2020 and awarded much later. But that kind of finished it off. So it was China, Thailand, Singapore, Malaysia, Laos, Myanmar, and Vietnam. So that is the DARPA Defuse geography, essentially, with Peter Daszak, Ralph Baric, Lympha Wang, Daniel Anderson, Xi Lee, Vincent Munster. No fear and cleavage site. Referenced fear and cleavage site. Baric put that back in there in this U01 grant. No fear and cleavage site. 3.2, 7.5, 3.4. You add it together, you get 14.1. And the original DARPA Defuse bill was 14.2. So this is DARPA Defuse being funded by Tony Fauci. And that's why he said he never seen that grant or funded that grant. I don't know what Fauci saw. He signs off on every grant. I believe him. He probably didn't know what he was getting into. He obviously didn't know he was getting into a fear and cleavage site and whatnot. But he did bring his own, lab into the fold, so that's his responsibility. because of, this fear and cleavage site by itself is pretty harmless, but this guy right here and his aerosolized agents are very harmful. It's the worst of both worlds. Fear and cleavage site meets his aerosolization. And that's what kind of led to the pandemic. I don't want to dive up too much into this stuff, but here you can see real quick the Creed FOIA, finally came through unredacted, like last year, meaning 2024. And you can see Daszak has taken essentially the what was it? Where was it? Let me go find it. Now I gotta go find it again. He has taken the old document right there. This is page 22, and he's resubmitted it here into the Creed grant. And a figure like this would be good. The KG is Kevin Olival, I believe, but instead of breaking it down by TA, maybe the country specific or A. See what they've done? They've they've taken the TA1 and the TA2, and they're now breaking it up into a new NIAID grant. So that was the backbone of the this is the draft Creed bid in June of 2019. So you got to bid these things before you get these things. You got to experiment with these things before you get these things. So we split it up. The TA1 team is over here on the left. The TA2 team is over here on the right. So it was pretty brilliant what he did. And Daszak's Defuse virology bid TA2 scored a 32. It was scored a 32 in November of 2019. That sounds like an interesting date, doesn't it? And who's all on here? Lympha, Danny, Ralph, his wife, Amy Sims. She was on DARPA Defuse. Broder's a new name with USU. That's how we got this email. US Right to Know had to go to Broder's inbox to get this email. That's because Ralph Baric won't give it to us. Lympha's part of Duke, which is a private university. Peter Daszak obviously is not going to give it to us. Super Porn, you'd find her on some of the U.S. predictones. All these other people right here are EcoHealth. They call it EIDRC, but in November 2019, they scored 32, and that put us right on the edge. Put us right on the edge of being funded. But Fauci down here is going to be making this decision, and that's November. So Fauci's Creed contractor Daniel Anderson was being graded by Fauci himself in November of 2019. That's when she left the Wuhan Institute of Virology for good and left Wuhan for good. So, what else do I have on here? This gets into some details that I want to talk about here. Daszak testified, whatnot. So, how did Ralph Baric get the RATG genome? It was from the R01 grant. Peter Daszak talked about a data dump from people like, Xi Zhengli of the Wuhan Institute of Virology.
Today, all the samples you have, that you don't know all the research, so you don't know that it is impossible for accidental release being the source.
Well, what we do know is, prior to the outbreak, we'd asked the WIV for every single SARS-related bat coronavirus sequence. Not only did they share the ones from the work we were doing together, they also shared.
But sir, this is the Latin paper that only goes to 2015.
Well, let me finish my point. They also shared data from work they'd done themselves without our involvement. So I think that was very important. If they had any other sequences at the time, surely they would have been in that data dump. And
that data dump was what was published in the Latin paper,
correct? Yeah.
So that only went to 2015. Did you ever request all of their SARS-related sequences and samples from 2016 to 2019?
I requested those data prior to the outbreak in something like November-ish or fall of 2018. We then went back to them repeatedly throughout the process, backwards and forwards. Any other data that came would have been requested in 2020. So yes, we did ask.
So the Latin methodology is wrong.
The data dump precluded RATG13. Xi Zhengli started to upload RATG13 to an NCBI database in June or July of 2018. She uploaded parts of it, then obviously the rest of it was just dumped here in November 2018. And someone like Baric was on the receiving end of that. So that's why Baric was added to the R01 grant, to give him access to Xi Zhengli's freezer. So this isn't like nefarious. Baric did the exact same thing with SHC014. Xi Zhengli shared it with them before publication. And Ralph Baric added her name to the 2015 Nature paper, talking about this novel chimera they created. So it's just science in action. It's unfortunate it resulted in a pandemic, but accident, people, keep the word accident in the front, the back, and the side of your brain at all times. No one set out to blow up the world. It's not some nefarious act by any means. But the creation of COVID was intentional, and the cover-up was intentional. You would do the same thing if you're stuck in their situation. You'd lie through your teeth because you think you could get away with it because everyone was focused on Xi Zhengli and the Wuhan Institute of Virology. But I don't want to get into, why we don't include her as a suspect. She published our ATG-13, which one of, Baric then wanted arrested for doing that. So this gets into this part right here. The fear and cleavage site was what everyone was looking for, and so was I. Everyone would come to me like, Jim, you're a lunatic like U01 DARPA Defuse because it doesn't talk about a fear and cleavage site. So it did. Deep in the document is a reference, Baric's reference. So this is DARPA Defuse at the top. This is the Fauci's creed at the bottom. So it's the exact same reference: computational protection of your cleavage sites, computational protection of your cleavage site. It got deleted in the final draft due to space constraints. So this is the beauty of DARPA Defuse. It was 75 pages. So what made your brain kind of just spin out of control was actually, the key to figuring this all out, because they had to put so much detail into DARPA's proposal, because DARPA was not comfortable with Ralph Baric's, genetic engineering, his gain of function research, but Tony Fauci and NIAID were. So they had to explain to DARPA what they wanted to do precisely down to like the genomic level. And, the younger DARPA crew was not as comfortable with the older Fauci crew. So it was always in my mind, it was hard to prove how that someone like Fauci would be comfortable funding something like DARPA Defuse. So to get further into the weeds, the human airway epithelial cells, which are not found in the one in virology, I've got into in a previous blog, go there for that, are found in Baric's University of North Carolina, and it was included in the R01 grant renewal. And the fear and cleavage site is required for in vitro growth of a SARS-2 fear and cleavage site. Otherwise, Vero cells is deleted immediately. So, and Daszak started on a post-pandemic was the Laos grant. So there's a Laos Binal 52 sample that could possibly involve this grant. I don't want to get into that kind of speculation. The U.S. military was there in 2017. But Duke and other people crawling around Laos. I mean, pretty much everyone was in Laos. This is. I don't know if I want to get into this right here, but I guess I will because we're here. So once you get your brain around the above, it's easier to follow what happened below. You can see HKU3 ARCOVs inside Fauci's NIAID grants, and that's essentially the SARS-CoV-2 genome. So this is more of a point for future FOIAs, for future truth seekers to go in there. I don't know if Baric will ever release his documents. He's never released a single email from 2018 to 2019. But in those emails is obviously Baric talking about a SARS-2-like genome called HKU3 ARCOVs at the time. So what was called HKU3S mix. You remember the HKU3S mix? So we got the 25%, diversity here. So HKU3, 25% different genome. So HKU3S mix is SARS-CoV-2. That's the SARS-CoV-2 spike. It's not like nucleotide level, but it's just the patent, the grant, the name for this novel backbone he's created. This novel chimera Baric's created. It was the SARS-2 genome, in other words, was sitting in 2018 documents, and it was also sitting in 2019 documents. So that's it. HKU3 ARCOVs, 25% divergent. So SARS-2 is 20 to 25% divergent from SARS-1. It gets really tricky. Nucleotide, whole genome is 20%. Amino acid, just the spike is like 24%. So it's around that little area. It gets really, really tricky. I don't want to get into that here, but this is the approximate signs on there, the squiggly mark. So this is the SARS-CoV-2 genome in Fauci's 2019 documents. It's sitting right there underneath our nose the entire time. Here it is in the R01 for the Laos bid. Then most importantly, here it is in the U01. So a lot of people run with this and question it, but I don't want to dwell on it too much. Once you understand the above, this will make more sense below. So and just more proof that the HKU3 genome was basically a placeholder, I call it. Baric used the HKU3 name as a placeholder for 2018 patents and 2019 grants. I mean, it's not, this SARS-2 genome is a work in progress. It's a pietà. It was his masterpiece that leaked from that Wuhan lab. But he recognized it when it leaked because he patented it in 2018. It's quite the story. Once you get into the details of it, it's one of the greatest stories of all time. So when you found out that Tony Fauci funded this research on U.S. soil and shipped it off to Wuhan for testing, and it leaked accidentally, I think it becomes one of the greatest of all time. I mean, it's one of the most incredible things, So Baric was very, odd in his post-pandemic, presentations. He does not talk about the SARS-2 fear and cleavage site. He literally, just doesn't want to talk about it. And what else was on here? Lympho explained we each had a part. So this is important. A lot of people, they don't understand the scope of work. They don't understand the division of labor. But Lympho Wang does a good job of explaining the division of labor. He's the bad immunology, essentially. This is Alina Chan asking the questions, but but someone like Shizeng Li does the bat sample collection, he says, and someone like Ralph Baric would do the fear and cleavage site. So it's a massive intercontinental research program. It's not hard to fathom. You get on a video conference like you do here, and you get along with your colleagues, and you do this probably on a weekly basis, and you touch base on what all you're working on. And each of you are doing something separate. Now, you couldn't do what they did, and they couldn't do what you did. That's the division of labor at work. It's kind of the way progress is made. You split up and divvy up tasks, and you specialize in what you do individually. And the collective whole is something better than the individual parts. So here's what Lympho said. You can learn a lot from Lympho, as did I.
Site from nature put into it. So my question for Lympho is, who proposed this fear and cleavage site insertion in the paper? Was it Shizeng Li? Did she propose to put the fear and cleavage site? So was it Ralph?
I mean, that's not, I don't know who proposed. That's not, the part, we each have a part, right? The Wuhan Institute was the field research, either bat immunology, and UNC is doing the virology, so that it's pretty obvious, right? Yeah.
No, but who proposed the idea? Because the idea says that we are reviewing our deep sequencing data. We've seen these cleavage sites, and we're going to put them into SARS viruses in the lab. So the question is, who had the idea and access to this data and could do it in the lab in a seamless way?
UNC, right? It's a UNC lab. This, I mean, the grant is very clearly.
It's University in North Carolina. In case
yeah.
So it is pretty obvious if you know what he's talking about. So this talks more about that 25% different genome being HKU3. So and here's another little thing that I'm going to talk about. Why I got it underlined here. So when Shizeng Li published RATG13, it was 4% different from SARS-2. And Baric expected it to be something less than 5%. These are Baric's words. This is his consensus sequence right here, consensus candidate. So Baric has created a novel chimera, a consensus sequence in 2019 that was less than 5% different than something like RATG13. It was so new and novel, Baric wanted it to not even be 5% closer than anything else. And so this is another, prediction from the 2018 DARPA Defuse document, essentially predicting something like SARS-2, a consensus sequence would emerge as less than 5% different. Lauspinol is also 4% different. So he was very specific with his verbiage and very specific with the way he did it. So, and that was, we already touched on this, I think, if not this video, maybe the next. It was exempt from dual-use research of concern. So this is a heck of a story, in the grand scheme of things. And here I break it up in the chapter. Read Murphy's letter. It's chapter 7 when I break it down. I also break down the DARPA Defuse document there too. So let's again, let's just keep going forward here real quick. If I can finish this up, it'd be great. These are the DARPA Defuse notes taken in March of 2018. And this is Baric, has all kinds of detail. Lympho has a lot of detail, Danielle technically doesn't, but she emerges in the Creed document as a person of editing interest. But here in 2018, the only time Shizeng Li ever responded to these guys was this one time in March 1st of 2018. Great, thanks. That's all she said in 1,400 pages. She responded one time to an email. And says great, thanks. She's basically just providing live bats for someone like Danielle Anderson and RATG13 samples for someone like Ralph Baric. That's all she's really doing. She's just a cog in the wheel, So it's kind of interesting. And a lot of people then come at me and say, well, where do you find the actual bat vaccine being talked about in the DARPA Defuse grant? This gets a little in the weeds, but this is Barrick's own words, in March of 2018. He down here, it is anticipated that recombinant spike glycoprotein-based vaccines harboring immunogenic blocks across the group 2B coronaviruses, sarbecoviruses, will induce broad-based immune response that simultaneously reduce genetically heterogeneous virus burdens in bats. That's a lot to digest. Thereby, just focus on this, thereby reducing disease risk in these animals, bats, for multiple years. So that's Baric's, convoluted way of saying, I'm going to zap the bat with my novel chimeras and reduce the disease risk in these animals for multiple years. So you see what he's talking about there, the HKU3. So not even Peter Daszak really understood what the heck. Clearly, I don't understand your details, and I don't even know if Baric fully understand what he's doing, But he's just throwing the kitchen sink, and he likes to create novel chimeras and test them on live bats. So that's that. That's essentially Baric, creating a novel chimera. There's some other stuff in here. Let me see if there's anything of any interest to us. I don't think so. Yeah, this is another myth of the DARPA Defuse document I want to talk about. So if we win this, this work will not necessarily come together, Ralph. I do want to stress the U.S. side. So essentially, Baric is when he's sitting here saying in the U.S. these SARS are starting under BSL-2, BSL-3, especially important for those who are able to bind and replicate. In China, it might be otherwise under BSL-2. U.S. researchers will likely freak out. Baric's talking about Ebola, Marburg, Hendra, and Nipah, kind of thing. He's comparing the BSL-2, or Peter Daszak was comparing the BSL-2 nature to this BSL-4, and Daszak's pimping, or selling the fact that their work on coronaviruses is cheaper, and Baric says, you need to replace that with BSL-3. We're still, though, the idea was that, because Xi Jinping even uses a BSL-3 with live animals, Baric got her to do that, to his credit, in 2019. But they're comparing their coronavirus work to more expensive Ebola, Marburg, Hendra, Nipah that has to be done in a BSL-4. So that's a bit in the weeds. But this is back into the good details of things. This is DARPA Defuse in 2018. This is NIAID Create in 2020. So you got Lympha, Daney, all the EcoHealth people. Baric, Tim Sheehan replaced Amy Sims. Amy Sims was on the DARPA Defuse grant there. So you got Lympha Wang, and Daniel Anderson was underneath them, and then you got Xi Jinping and all the wanted to do virology people, and you got EcoHealth and all them. So there's that. And just to drive home this point, I've played this a couple of times. I guess I'll do it again. I really want to drive home the point that Tony Fauci himself was involved in live bat research. This is like a challenge trial of EcoHealth Alliance, Peter Daszak Epstein, with Fauci editing it on January 8th of 2020. So that is in essence Fauci being involved in something very similar to something like SARS-CoV-2, or something like DARPA Defuse. So again, DARPA Defuse is all about bats. It's not about humanized mice. I think I did that in the previous video, but I'm going to do it again here because you hear a lot about humanized mice. It's only mentioned 8 times. So in DARPA Defuse, bats are mentioned. I think bat or bats is 300 some odd times. So there's that. There is something interesting with the batified mice. So this came out early, like what the hell is that? I don't know if I still know what a batified mouse is, but it's something Lympha Wang has produced. But it's also found in Fauci's creed. So here it is. They call it bat mouse model right here in the middle. So it's just another hangover, effect between DARPA Defuse and Fauci's creed. And Fauci's creed project, it was posted in March. They had until June 2019 to submit it. And then the review is going on in November. So this is the TA2 in DARPA Defuse right there. So yeah, here's another post if you want to kind of just dive into the details of it. This is where I find this is where I put it together. In November of 2022, I know COVID is a contagious bat vaccine developed in Fauci's biodefense lab. Why? Because Fauci's own biodefense lab in Montana has won the DARPA project. The winning DARPA team in Montana was a lot more interesting than the losing team in Wuhan. So in 2018, Fauci's Montana lab, it's his lab, by the way. I'll show you a video hopefully here. Maybe not because I have it closed, of Fauci's lab in Montana, essentially kickstarting, this DARPA winning project. So you can go through here and you can see Tony Fauci's aerosol specialist showed up at this conference. There he is right there, Vincent Munster. That's Vincent Munster, who's been added to the U01 grant right there. So that's him of Rocky Mountain Lab. That's him in a spacesuit. So he is the creator of COVID-19. I've asked him for an alibi. He hasn't given one, nor can he. So, let's not feel sorry for him. Here he is hanging out with Peter Daszak, in October of 2019. Peter Daszak recently locked his account, so it's kind of nice to have this, time-stamped here. This is Vincent Munster, showing off his fancy bat cufflinks. He was, he's kind of like the BSL-4 Batman of NIAID. He is studying live bats in that Rocky Mountain lab and giving presentations here in Germany. In October, hanging out with Peter Daszak. I always thought that was an interesting date. And he does this one at the bottom called a transmissible vaccine. It's you inject the needle into the bat, into the animal, into whatever animal it is, and it spreads autonomously to the next one, like a virus. It's a contagious virus that acts as a vaccine. Whereas Ralph Baric up here, this is why I've eliminated the USGS and all the other outfits from the list of suspects. You can see basically the USGS role is like Rocky here. They will optimize delivery of immune modulating, biologicals they call them, vaccine delivery work on wildlife, including bats. So that's this up here. She was going to do this method right here with Ralph Baric. The USGS and Ralph Baric wanted to do this method. You spray the bat as it leaves the bat cave. And the bat kind of just licks it off, the fur from it. It's called a transferable vaccine. So it's 1-to-1 or 1-to-many, where this is kind of many-to-many. So you can see just the way the R0 just like a ski lift, it just takes off quickly. This is more effective, obviously, for coverage. But if you just look at it for like 5 minutes, not even that, 5 seconds, you realize this one's dangerous. This one isn't. This one, it can't jump species. It can't, infect every mammal on the planet. This thing can. It was a dumb idea, basically, being promoted by this guy right here. That's what he was working on. He's an aerosol specialist, a transmission specialist. He can take a pretty benign inert agent, like Ralph Baric's HKU3R Cove, and he can turn it into an airborne, agent. Not for nefarious purposes. This was an animal vaccine. It was an accident, but let's have an adult conversation about it. This is what he was doing when he won the DARPA project. This is why he was so cheap. So Peter Daszak was 14 million. Vincent Munster was like 8. Peter Daszak's phase 2 was like, what was it? It was like 5.5 million. I mean, I'm guessing his was, less than $1 million. I mean, there's not much development cost once you get that needle, formula right. Again, that needle can be shipped around the world on cold ice, or the cold chain as they call it, and it's just a vaccine. It's a single jab into a single mammal at a single point in time, and it's not going to spread mouth to mouth to the next mammal next to it, kind of like us humans. So that's what SARS-CoV-2 is, a contagious vaccine. And here's somebody who was kind of blowing the whistle. Transferable vaccines that can jump once from an initial animal to others, then stop, are almost as effective, easier to develop, and pose fewer risks. Save playing with pandemics for when we can defend against them. So he's saying the transferable vaccine, which is what Ralph Baric and USGS and Peter Daszak proposed, was safe. And I agree, but this transmissible vaccine was not safe. It's pretty darn dangerous. So that's a fruit bat. Hammerhead, I believe, and what they call them. But that's Vincent Munster right there. He was the Batman, basically, taking a blood sample from this one. So this is a good little visual, the difference between a transmissible on the left and a conventional on the right. Watch the transmissible one spread quickly. This is a self-spreading vaccine lab in England. I mean, this was all the talk, all the rage in 2019. Because it was, look at the endless potentials to preempt the next spillover event. It was just huge. So the deer mice were used as transmissible vaccines. And just drive home the point that Rocky
administrators, the secretaries, the animal care staff, and the maintenance staff can come together and sit down, talk, have a cup of coffee, and discuss the work.
The Rocky Mountain Laboratories had a critical mass of talented scientists who were in the same place, interacting with each other, and fully capable of studying everything from the basic science to the ultimate applied development of countermeasures.
So that's Fauci's biodefense lab in Hamilton, Montana, where they study things like deer mice, which is also a SARS-2 reservoir. So this is how COVID spreads in humans. This is how it was ideally going to spread in bats. Bats and humans, unfortunately, have a similar ACE2 receptor, a similar upper respiratory tract temperature, similar furin in our mammalian cells. Bats also have mammalian cells, so they also have furin in them. So when they were targeting the upper respiratory tract of a bat, they, as Vincent Munster, he unfortunately was also modeling the vaccine to transmit in us humans. So, this is essentially the airborne transmission. It was always a wonderment, why is it 100% infection rate but a 99% survival rate? Well, it screams vaccine. It screams transmissible vaccine. Once you get your brain around it, the immune evasion, the aerosol transmission, upwards of 60 feet, he does it by doing the microns. Like he gets down to like 5 or 6 microns with this thing by using small animals like Syrian hamsters. And then he extends the cage, and extends the range that it can do. He was just trying to get the aerosol to work for bats, tiny little lungs, tiny little mammals, tiny little creatures. But for us humans, it just amplifies the R naught and the spread. We're just some of the biggest, mammals on the planet in the grand scheme of things, when it comes to something like this, So, yeah, unfortunately, our human lungs became bat lungs in this whole thing. So this is a good little piece, I think. It gets a little deep into the weeds, maybe. But this is the way Munster operates. This is replace the ferret with a mink. That was one of the other transmission models he used. There's no mink found in China, but they are found in his Vincent Munster's BSL-4 lab. But he basically just creates a positive pressure flow, or uses a negative pressure pump over here, and just draws the air from one infected mammal to the next as he's training the pathogen to spread. Not through nose, not through contact, but through air, and he'll extend the distance between the cage to extend it to spread further. He's not doing anything mean, or nefarious. He's just trying to get it more transmissible amongst the mammals. So that's kind of, I think where this story, on this post ends. And this is why we're in Wuhan. Vincent Munster had to go to Wuhan to test the vaccine. It was the only place on the planet with access to Chinese horseshoe bats. Technically, these aren't Chinese horseshoe. They're myotis, but the idea was that there are live bats inside the Wuhan State of Virology.
At some point, we should keep a list of all the conspiracy theories that turned out to be not just Australia. We know it's a lie.
There are bats in the lab, so that's where that's going. Fauci here is defending himself. I only funded in vitro work and in the sub-grant. So that he's holding up 2017 paper with Vera cells that would delete the furin cleavage site even. But it was an alibi that doesn't hold up since he funded DARPA Defuse, and there's Xi Jinping in the NIPA conference in 2019 in Singapore, along with Danny Anderson, Ralph Baric was the only one not there, Vincent Munster, all the DARPA Defuse team was there, Peter Daszak, Lympha Wang. So this is a good place to start. This is when I knew COVID was a contagious bat vaccine developed in Montana. It was mainly because of this FOIA by U.S. Right to Know in January of 2021. That was even before I got into the lab leak debate, to be honest with you. And people like Cy here were issuing FOIAs on the DARPA preempt program. They were working on, Rocky Mount Lab was working on self-disseminating vaccines. So the Rocky Mount Lab and Vincent Munster had won 2 of the 5 DARPA grants. Peter Daszak and Ralph Baric lost because of their relatively safe but expensive technology called transferable. But Vincent Munster had this scalable vectored vaccine. Remember, scalable is what DARPA wanted. If I still have that open, I don't know if I do. I thought I did. But if I do, where is it? So scalable. Scalable. That is what they want. Scalable. Vincent Munster delivered, what they wanted. Scalable delivery methods, So that's why, in the scalable delivery method, you'll see it talked here. This is the July 2018 meeting minutes with Vincent Munster of Rocky Mount Lab and the winners. He's talking about scalability. The scalability. He's using a VSV. Baric brought an LIB to the table. So some people get kind of lost there. But they're developing a scalable vectored vaccine for, this was Henipa viruses like Nipah in Australian bats. But the virus in the bat, don't matter. Well, the bat does because he's using Egyptian fruit bats, a reservoir host for SARS-CoV-2. So deep into the documents, you'll find Vincent Munster using an Egyptian fruit bat as a surrogate, a transmission model for the Henipa viruses. And the Egyptian fruit bat didn't work. He told DARPA that in 2019, but it did obviously work for SARS-CoV-2. So where is it? I think it's right here. So Rosetta's I can't even pronounce the Latin name there. Egyptian fruit bats could reflect a reservoir host status for SARS-CoV-2 and present a useful model, although the species is certainly not the original reservoir SARS-CoV-2 because these bats are not present in China, but they are present in Fauci's Rocky Mountain lab in Montana. So, yeah, we're going to kind of get a little deeper into the weeds here. Hopefully I don't lose you. But, once you get your brain around bat vaccine, a lot more will start to make sense. So, essentially, DARPA has told NIAID, or DARPA has told Baric, no, you're too expensive. So what Baric's done now is gotten together with Vincent Munster, the winner. So the winner of DARPA Defuse has teamed up with the loser of DARPA Defuse here in late 2018. This is after DARPA said, no, you're too expensive. Now, all of a sudden, Fauci has apparently merged the two, or Vincent Munster has merged the winner and loser underneath his DARPA, or underneath his ongoing DARPA projects. They're infecting Egyptian fruit bats in 2018. So the key was this is the DARPA Defuse grant. They were going to inoculate bats with WIB1. I could not figure that out until this virologist in London did.
Any of that spike engineering, recombinant virus work anywhere else other than UNC. WIV's role was to sample the bats, sequence them, make recombinant proteins, look at the ACE2 interaction.
Infect live bats. Infect live bats.
Oh, well, that was
the IV.
But also, what were they going to infect the live bats with?
Well, I don't know, the viruses that they were studying.
They were going to infect it with WIV1, and they were going to do it as a proof of principle. But that
DARPA grant
was such an aspirational part of the grant. There was no way that anyone's got that far yet.
Well, they had gotten that far because obviously they're doing it here. They're infecting live bats with WIB1 in Rocky Mountain Lab. So this is Baric continuing on with the live bat research proposed in DARPA Defuse. He wanted to inoculate bats with the WIB1 genome. So that's kind of where the DARPA Defuse project goes forward. And here it is. This is the U01, i.e. the TA2 in DARPA Defuse. I know it gets confusing. Just stay with me. That's this grant right here. But inside that U01 grant, Rocky Mountain Lab, where Peter Daszak and Ralph Baric have ongoing collaborations, are going to be in the receiving end of some genomes and animal samples. So Rocky Mountain Lab, like North Carolina, is on the receiving end of Chinese genomes and samples. So they've gotten the band back together, but now they're going to use a transmissible vaccine. So again, I know I talked about it so many times, I'm going to keep talking about it till the end of days. That is Xi Jinping basically announcing that no one in her BSL-2 lab inserted that pure cleavage site. It's a bit preposterous to even entertain that. So but she, when Xi Jinping published it, she said, not my lab. And so, it's pretty obvious what lab she's probably talking about here. Her lab, her BSL-4 down the road, where she was a pretty important person, She's being talked about here by the NIH people. There's a collaboration at Duke NUS who will act as a consultant on the project and provide her expertise in serological testing. So she does antibody testing. And you would think humans, but no, she's a bat immunologist. She does it in bats. So that's what she does. She tests for antibodies in bats. It involves needles. There's all kinds of ways for this thing to get out when you're involving that much needles, that much blood drawn, that tiny of a bat to experiment with virus characterization and PCR detection of viruses in bats. She's a bat immunologist. She said as much to her credit, to her defense, here early in the pandemic.
Outside central Singapore.
I've talked about this in another video, but I just love it, that little gun drawing person, don't come near here. I don't know what that is, backup generator. Maybe it's the breeding colony for the bats. That's Duke NUS right there.
Research lab where scientists have isolated the purest form of virus from.
So she's going to handle a live bat.
Bats gathered in China for food. A vet here captured one. And in a containment cage, she showed it to me.
And why bats? How confident are you that the coronavirus came from bats?
So I'm probably 90% confident that the new coronavirus came from bats.
So she's 100% confident because, the SARS-2 genome was probably inside of a bat, inside the Wilderness Virology, inside her BSL-4 when it accidentally got out. So that's what the experiment was, to infect, something like SARS-CoV-2 into bats. And again, how do I know that? Because SARS-CoV-2 was working pretty well, obviously, in Egyptian fruit bats. So the idea was to extend the test extend the test to the one Institute of Virology. So, yeah, and to go on here, a lot of people have a hard time with the bat vaccine being SARS-2, but, good luck proving otherwise. SARS-2 pathology equals bat immunology. I love that line, which only apparently I appreciate, but, whatever. So, you got to be comfortable with the fact that you have a 65 million year old immune system. Bats are not birds, they're mammals just like you and me. Long arms, opposable thumbs, tailless kind of body, kind of forming, way up in the trees or way back in the cave, way back in the day. So, that's what Baric was studying here in 2017. He's talking about bats' immune systems in particular. So it's pretty interesting stuff once you get into it, to be honest with you. All these weird little pathological terms from SARS-2, inflammation, orphate, interferon, MHC1, NLR3P, cytokine storms, B and T cell. These odd terms are all well described in the Western bat vaccine literature. Remember when T cells were politicized in 2020? They were studying American bats and deer mice in 2017. MHC class 1, Baric and Munster studied that in a Montana paper. Spreading back super spreader events, we all know that now, but vaccinologists were dreaming about super spreader events in 2019 with the transmissible vaccine research. Even the weird asymptomatic nature of this virus, called immune evasion, asymptomatic spread, was designed, featured, not a bug. As Major Murphy told us, a good vaccine does not generate symptoms. Self-disseminating vaccines create a moral hazard for immune evasion. So basically the idea is to avoid the immune system so that the vaccine can keep spreading and shedding, just like SARS-CoV-2. So the bat virus can jump species, so can a bat vaccine, made to spread like a disease. If you replace the word virus with vaccine, just stay with me. It's getting kind of weird, but bear with me. If you replace the word virus with vaccine, infection with inoculation, switch she with Danny, replace humans with our distant ancestor bats, everything will make sense. This animal vaccine was pre-adapted for American lab animals being tested on Chinese lab bats. So, Baric, used the word national security as live attenuated vaccine. That's this one. A lot of people push back on me because they're like, Vincent Munster was using a VSV, here, wasn't he? And I go, yeah, but, that's here in October 2018, again, after he's lost DARPA Defuse, Baric's gone in there and basically admitted he has to create a bat vaccine based upon where is it where he says it? I've already lost that spot apparently. So I don't need it. But when Baric's going in here, he calls this thing a national security paper. That's the way he saw it, national security, and he's talking about an attenuated vaccine. Attenuated. So, and in particular, he's talking for a reservoir species such as bats. So furthermore, reservoir species such as bats. And it's creating a, he's even referencing the Moisheng mine shaft. That's where this is the paper that referenced the 4991 sample renamed RATG13 later on and published in 2016. So Baric's got the RATG13 sample for this paper. So it takes a little while to get your brain around all the craziness in it, and even inside of it is a TRS, a transcription regulatory sequence, I believe it's called. And right there, UGG, UCGC, you'll find that in the RATG13 sample, pretty much at the ORF1A sequence. And you'll also find it, it's moved slightly in SARS-CoV-2, but you'll also find it in the exact same spot in Laspinol 52. So this what he calls CRG7 TRS is essentially him fiddling with the RATG13 and Laspinol 52 genome in 2018. So this is the live attenuated vaccine beats Baric's, transmissible vaccine. So I think that's basically it as we wind this down, just to kind of summarize here. I think we can see Ralph Baric, talk about things like intracellular proteases. I think is something I want to talk about, if I can spell it and find it. So intracellular. There it is. So these are Baric's notes during a Ralph obviously, is being talked about here, a proteases cleavage site here. Either extracellular proteases or intracellular proteases have to clip the protein once or twice so that the virus can fuse to the cell membrane. Viruses can't enter without proteases. In the lab, if proteases is added exogenously, strains that otherwise would not enter cell are now able to. So I don't even have the paper open, do I? Well, yeah, I do have it open, thank gosh. So intracellular, there it is. So right here in 2018, Ralph Baric has teamed up again with, this is so deep, bear with me. Ralph Baric has teamed up with Vincent Munster in 2018 to infect Egyptian fruit bats. And, this is obviously Baric's words here from DARPA Defuse being carried over into this, document. It is possible that the Wuhan glycoprotein, they're publishing a negative result, which is kind of odd in virology, but they're telling you why, because they want to add the furin cleavage site into the genome to get an infection of the Egyptian fruit bat. So this is kind of their way of saying, fund the damn DARPA Defuse program, Fauci. So if they add, intracellular proteases, they can get in there. They can get into the bat. They can get into the bat cell. And it's obvious when they're referencing 12 and 13, they're talking about a furin cleavage site. You'll even find the infamous Drosten on here. So, this is a collaborative effort where they cross-reference each other in these semi-incriminating documents. And, at the bottom of this document in 2018, they want to test the final product with the furin cleavage site using Chinese horseshoe bats. So that's where Daniel Anderson and the BSL-4 come in. You had to go to Wuhan to test on Chinese horseshoe bats. They had Egyptian fruit bats. They have Jamaican fruit bats. They have all kinds of old, or what I should call New World lab animals in the in Rocky Mountain lab, but they didn't have the old world tiny eating horseshoe bats. Those are only available in Wuhan. It was the live Chinese bat capital of the world, and that's the only reason Wuhan's a story. You had to go to Wuhan to test it, and there's only one person on the planet that could test it. Bless her heart. She was a bat immunologist with access to live Chinese bats inside of a BSL-4, which they thought was safe. So the end results here are published. The Egyptian fruit bat now went from not being a SARS-1 model. WIV1 is basically SARS-1, if I haven't said that. It's a stand-in for SARS-1. So SARS-1 does not infect Egyptian fruit bat, but SARS-2 is now, Egyptian fruit bat is now a SARS-2 reservoir host. But the Germans here that published this were, obviously these bats aren't present in China, so they're not involved. Well, obviously those bats are present in Rocky Mountain Lab, which have not published their Egyptian fruit bat results. And so I'll get into the smoking gun email in another piece, but I think we'll leave it there. I think this is enough to basically show you where we started off as, the man who funded DARPA Defuse was, he always used the word molecularly impossible. So let me finish off with that line because I love it so much. It's just fun.
Was responsible for funding this type of so-called gain-of-function research, but we don't know. Do you think that your early resistance to that was ill-placed at the time?
Neil, I'm sorry, this is revisionistic history. I have always had an open mind and have said right from the get-go that we should keep an open mind as to the origin. But as more and more data came in, including now recent papers from highly qualified virologists from many different countries, have shown that it is much more likely that is a natural occurrence. Does that mean we know exactly the origin? No. And that's the reason why we, myself included, right from the very beginning, Neil, have kept an open mind about that. You can keep an open mind and still have an opinion of what the most likely origin is, and a large group of highly qualified, unbiased scientists feel that it is most likely a natural occurrence. With regard to what the NIH did in funding research, the viruses that the NIH gave a very small amount of money, $120,000 a year, to do surveillance studies in China, the viruses that they were dealing with, anyone who knows even a little bit about virology will tell you that it would be molecularly impossible for those viruses to have turned into either accidentally or deliberately SARS-CoV-2. And yet people seem to discard that, and they go on with a wide variety of conspiracy theories. Bottom line, I always keep an open mind.
He's a brilliant man who knows how to lie, a half-truth basically. So she was studying the WIV1 samples, essentially the SARS-1 type samples basically. But and here was the HKU3R cove that was inside. So that is quite the way to end this, I think. The SARS-2 genome was in Fauci's 2019 documents. So 2019 R01, the U01 is the SARS-2 genome. That is 25% divergent from SARS-1. and SARS-2 is about 25% divergent. I'm not going to get into the details about that. So if Fauci funded DARPA Defuse, and so do we, as we seek the truth here. So I'll end it there, guys. Thanks a lot. I think we're going to do it. I may go into part 3 later on if I feel like it, but I think it's enough for now. So if you got any questions, let me know, and I'll see you in the next one. Bye.